Okay, so in this video, we're going to begin the very first section of chapter 3. So we're going to talk about cells, and we can see this really cool picture of a stained cell. You can see all the different parts that are stained different colors with the fluorescent dye. Um, so you can see the microtubules, you can see the chromosomes, you can see the cell membrane in this picture. It's a pretty neat picture. And we're going to talk about all those parts and a little bit later on in this chapter. We mentioned basics about cells previously when we talked about the different organization levels that you need to know for your test. We mentioned that they are the smallest unit in the body that is alive. Remember that not everything that is in our levels had all the properties of life. Not everything could grow, not everything was able to reproduce. Since its environment, we talked about molecules like water, glucose, those kind of things, those aren't alive. Viruses aren't alive. They can't do all the properties of living things that cells can. So they're the smallest unit that is alive. So the smallest living unit. They could not be examined until we had the microscope. So we knew that there were these small things doing stuff. We were trying to figure out, how contagion's happening, all this stuff, but we couldn't really have the hardcore proof in front of us until we could see it with our own eyes. So we could not actually look at these guys until we had the microscope in the 17th century. And debate is still around on who came up with the first microscope, but that is a debate for microscopy chapter in the microbiology class, which you will take if you were in health sciences later on. We talked about the term homeostasis previously, that you had a happy, steady state. It was a steady state of whatever that is, temperature, ion levels, whatever. You need to maintain homeostasis, and cells do that. And they are going to coordinate activities that allow homeostasis to happen at even higher organization levels, such as multicellular creatures like we are, where we have a whole entire organ system dedicated to that function. So definitely know the 3 tenets of cell theory. It's a lecture quiz question. It's always a test question on there. That everything that is alive is composed of at least one cell, if not more cells. That they are the smallest units that form all the vital functions. You can't go any smaller than cells. If you had a nucleus, for example, that doesn't do everything that's vital for life. An organelle is not going to be where it's at. Water is not going to be where it's at. We actually need a full-blown cell, and that does all the things that things are alive. All the properties of life are in cells. And all new cells are going to come from come from the division of pre-existing cells. Mitosis, steps of mitosis. We need to know them, love them. Those are always test questions, and you can't get one cell just magicking out of thin air, right? We talked about cellular reproduction, mitosis, binary fission. That's where we're going to get new cells. Cells are going to differentiate. So the cell in that make up the cells that make up my hair are the same as the cells that make up the skin, same as the cells that make up my eye. And in regards to DNA, they all have the same DNA. It doesn't matter what cells are in your body, unless we're talking about germ cells, germ cells like sperm and eggs. All your other somatic body cells are going to have the same DNA, but the genes in that DNA get turned on differently. So it's called cell differentiation. Because again, all the cells come from one cell, and that one fertilized cell is the zygote, where the sperm and the egg come together. Sperm's nuclei fuses with the egg's nuclei, and then we have the zygote that is now gone from haploid and haploid to diploid zygote, and it's got all the genes. That's the one cell, the totipotent cell that does everything and produces everything. So we all come from a single fertilized ovum, the zygote, and then it's going to divide. Cleavage happens within hours of fertilization. The cleavage is the division of the cell. So it goes from 1 cell to 2, and 2 to 4, 4 to 8, on and on. So as it starts undergoing cleavage, those cells are going to differentiate into different types of stem cells, and those will differentiate, and those will differentiate, until eventually you have all different types of cells. You're going to have cells coming from 3 different germ layers. You will need to know these germ layers. It is ectoderm. Ecto is going to be on the outside in general when you think of that germ layer, ecto, outside. So that's going to definitely include your skin. It's also going to include your nervous system stuff, which will make sense a little more when we go into reproduction in AP2 and you see literally how the um, zygote develops into a human being. You'll see how that central nervous system is linked in there with the ectoderm germ cells because it two forms. So we got skin cells, we have neurons, and our melanin that gives us coloration or lack of coloration, your pigment cells, all that's ectoderm. The mesoderm germ layer is in the middle. Again, anytime we use that in term, meso, median, it means in the middle. So the mesoderm is going to be the stuff in the middle, and the ectoderm is going to be the stuff on the inside. So I was telling y'all that we're kind of like a giant donut with a hole that runs all the way through us, the GI tract, your alimentary canal. So things that are linked to GI tract would be considered your endoderm. So that's going to include your GI tract, your accessory glands off of that thing, like your pancreas, your lungs, certain cells in the lungs, not all of the lungs, but certain layers of the lungs are included in endoderm. Mesoderm is going to be everything in between endo and ecto. So mesoderm middle is going to be your blood. You got muscle in there. Bones, all of that's going to be mesoderm. So cells will differentiate. When we differentiate, they turn on the genes that they need on that cell to be turned on, and they leave the other ones off. So they will differentiate, they will grow and look differently, even though they have the same DNA. We can see the different parts of the animal cell here. We are going to go into all of these different parts in the later sections. You will need to know these parts, know the parts of the animal cell, understand what they do. We needed to know the parts and recognize them in the lab, but we definitely need to understand what they do, what their job is, what their role is within the cell, because that's a large portion of the upcoming test. It's going to be a matching section. And so you'll need to know, the nucleus is going to house the nucleolus, the genetic information, the chromosomes, and it is in charge of coordinating all of that for the rest of the cell. So that's, what is it? What does it do? What's its function in the cell? So we will go through those later on in this chapter, and you will need to know those for the test. So I know most of us have covered cell parts maybe in as early as grade school, but we definitely are going to need to know cell parts for this class in this chapter. Moving on, let's go through the first section. I think we can knock that out. So the cell membrane is going to function to keep things in and keep things out, right? We don't want everything coming in, and we don't want everything inside being able to leak outside. That'd be bad too. So we are semi-permeable. It's going to help isolate the cell, what's inside, and what's outside. It's going to regulate what happens with the external environment. It helps us allow us to be sensitive to what's going on out there, because we said, again, properties of life, you need to be sensitive to what's going on outside. It's going to give the structural support to the cell. And we mentioned that there's going to be a plasma membrane. It's made out of phospholipid bilayers as well as cholesterol that helps give it structure. As far as lipids, it's going to give it isolation. And there are going to be a lot of proteins in the membrane. So here is a picture of the plasma membrane. You might also see it called the fluid mosaic model. Some textbooks call it the fluid mosaic model because the little proteins in here, they're not all locked in place. Sometimes they literally shift within the membrane. And so that's why it's called the fluid mosaic model, because those things aren't locked permanently where they are. They kind of switch around and float through. So again, what's its job? It's going to control things in and out of the cell. Ions, sodium in, potassium in, sodium out, potassium out. Who's coming in? What's coming out? We're going to control that. We'll talk about those with action potentials. Nutrients. Every cell is going to undergo cellular respiration. One of our favorite nutrients is glucose, so we're going to make sure that we bring that guy in, right? But we need to make sure that we're not bringing everything in. So we're going to control entry. We need to eliminate waste. We bring in glucose, we undergo cellular respiration. We need to get rid of our waste, and one of those wastes is CO2, so we want to get that stuff out of the cell. And we need to release any secretions that maybe that cell is in charge of making. We mentioned hormones and endocrine and exocrine and making chemicals. The cell's in charge of making those, right? And it's got to get those out. So it's going to be in charge of releasing secretions, like releasing insulin or glucagon or whatever chemical that needs to happen outside of the cell. What's it made out of? It's made out of 3 basic macromolecules. So we learned the 4 macros last chapter. The 3 macros involved in the plasma membrane are lipids. So we've got a phospholipid bilayer. That's these guys, these little red heads with the dangly tails. That's the phospholipid bilayer right here. And heads, remember, are hydrophilic. They liked water, so they face the outside where the water is at, or the inside of the cell where the cytoplasm is, and the cytosol, liquidy stuff. The tails are hydrophobic, so we tuck the tails and they face each other. So the tails are tucked inside right here. And these guys are phobic. These guys are philic. The other macro that we're going to have. So we have phospholipid bilayer. These yellow things are also a lipid cholesterol. It's going to give us structure. So all these little yellow guys right here and right here, these are cholesterol. Second macro, we've covered lipids. Proteins, there are so many proteins. So we will need to know the proteins and what they do. Sometimes the proteins are just on an inside, or they're just on the outside. They're on one side of the bilayer and not both sides of the bilayer. If they're on one side, it would be considered peripheral. Other times they go all the way through, so it'd be integral protein. We'll go through these more in depth in just a little bit. Then the third macro that's involved in the plasma membrane is the sugar, the glycocalyx. So glyco means sugar, that we already learned in chapter 2. And so glycocalyx, sugar can be attached to a protein, glycoprotein. So we can see the sugary molecule here, and then here is our blue protein. So this is a glycoprotein. Or the sugar can be attached to the lipids, the little red guys, the phospholipid bilayers. So this would be called a glycolipid. Altogether, the sugar molecules on the outside that are bound to structures in the cell membrane is called the glycocalyx. And if you want to, you can check out this animation. It explains a little more thoroughly and in depth the plasma membrane model. Here is a close-up picture of that phospholipid bilayer. Remember that they had hydrophilic heads up here, hydrophilic heads, and they had hydrophobic, didn't like the water, tails. The membrane, depending on where it's at, sometimes the membrane will have lots and lots of wrinkles in it. It may have microvilli, so instead of just being flat across, we might have all these different layers and wrinkles and all this stuff in here. And what that does is it increases surface area. So say you were talking about the GI tract. What's the function of the GI tract? Remember, we need to know all the different systems and what their function is. Even if we haven't gone into depth in that, we'll go into depth in AP2. But the function of the GI tract was to break down the food, right? And then you got to absorb it. So to absorb it, the more surface area you got, the better. So our intestinal cells are going to have this microvilli, this increased surface area, so they can soak up all the good stuff. So these are microvilli, and you can see there are all these infolding layers to increase the surface area. On our outline, it calls them, quote, finger-shaped extensions, microvilli. So those might be on your upcoming tests as a matching question, and you need to know what microvilli are. Finger-like extensions of the cell membrane out there to increase surface area. All the different proteins. So lipids were the first major protein that include the phospholipid bilayer and the cholesterol that's going to give the cell structure. Then we have the proteins. There's so many proteins. So we'll need to know the different types. Again, peripherals were just on the outside or just on the inside, and integrals integrated into both areas, both phospholipid bilayer, it went all the way through on both sides. Out of those guys, we have anchoring proteins, and they do just like they sound. They anchor it to this structure, whether it's anchoring it to another cell nearby or something else, but it's going to anchor it inside, maybe bind it to its own cytoskeleton within the cell, or it may help anchor to a cell next door. Recognition. So these guys are going to be detected by the immune system. So we're going to go into the immune system in AP2, where we'll talk about there are cells patrolling around here, white blood cells patrolling around your body all the time, looking for things that don't belong. If you are not supposed to be there, then that white blood cell is in charge of taking you out. So we have proteins that are out there saying, I am you, I am self, I am part of you, don't eat me. And those are recognition proteins. They help with that. Receptor proteins. So we are going to talk about receptors and ligands. Ligand is another fancy name for chemical. It's just another term for a chemical out there, an ion out there. And so the ligand is going to come along and it's your key, and your receptor protein is going to be your lock. And remember, we said protein 3D structure was super duper important. If your lock if the key doesn't fit in the lock, then it does no good. So it recognizes very specific things. So those are receptor proteins, and they are key to whatever they are built for, whatever they are looking to recognize. So those are receptor proteins, and it could be a really big chemical that it's trying to bring in, or it could be a small one, like just sodium or something like that. Carrier proteins. These are going to do just like they said. They will bind something and bring it across. So we're going to talk a lot about sodium and potassium in this semester. And so in the cell, your body is going to keep one ion in and one ion out. And so your cells are going to want to keep potassium in, and it's going to want to push sodium out. It's going to push it against the gradient, which we're going to talk about gradients and transport in a little bit. So it's going to want to keep things in and other things out. It wants to increase the level of potassium inside the cell, and it wants to increase the level of sodium outside. So it's constantly pushing sodium out. It's constantly keeping potassium inside. But sometimes when we bring things across, things are really big and they need a best friend to come with them to get in the door. It's like trying to get into that exclusive club and you've got the friend on the inside. So sometimes you need to bring a buddy with you. So glucose sometimes brings a buddy with him because we need glucose in the cells for cellular respiration. Sometimes glucose brings sodium with him to get in the door, to get inside the cell. And that's fine, and glucose gets in because we need glucose, but the cell likes to push all the sodium back out the door. So sometimes we have carrier proteins that will bind the molecule and shuttle it across to the other side, and it will carry it. Other times it's a channel, and that channel can be wide open, or that channel can be gated. It just depends. And that gate could be open or gated. They could be ligand gated. They need a chemical before they'll open. It could be wide open, like our ADH is going to, the hormone in your body, when we get into AP2, we'll talk about ADH, and it releases ADH into your bloodstream. It's come from your pituitary gland. And when it hits your kidneys, it's going to tell your kidneys to put in more of these little openings called aquaporins. And they look like this. And they are designed to open up and let more water in. So you will absorb more water into your body and you will be less dehydrated if ADH is in your system. Putting these aquaporins in, these wide open channels for water to flow through back into the cells. Another type of gated channel is a voltage gated channel. And we're going to talk about those with action potentials later on, that there's going to be a difference in charge between the outside and the inside of the cell. And that difference in charge will change depending on if you let ions in, because again, ions are charged particles, right? So if we change the ions that are inside versus outside, that's going to change the voltage in the cell. And that voltage change, once it gets to a certain level of millivolts, can open up a channel. So these are channel proteins. Again, the carrier proteins are going to physically, like 3D touch and hold it and shuttle it across with a carrier protein. Doesn't matter how much stuff you have outside the cell. If you increase the concentration, it doesn't matter because you're still limited by the number of carrier proteins you got. They can only bring in so much at a time. So it doesn't matter how much concentration there is. It's not going to increase absorption when you're limited physically by the proteins in the membrane that can only bring so much across at a time. Carrier proteins. The glycocalyx is going to be that sugary substance The sugars, again, they can bind to proteins or they can bind to lipids. Glycoproteins and glycolipids, what do? They also help with cell recognition. So we said proteins have the ability to meet up with the immune system and tell them that I am part of you. Same with the glycocalyx. They're going to help with cell recognition. I am part of you. I am self. Don't attack me. They are going to help to bind to structures on the outside. And they also help, sugar's going to help lubricate cell surfaces. We mentioned how water was a great lubricant. Glycocalyx is going to also help lubricate cell surfaces. All right, so that is going to finish that first part of section 1, and I'm going to finish the rest of section 1 in the next video because we're going to talk about membrane transport, and those things tend to take a lot of time. So I'm going to finish that part of section 1 in the next video.